I'm just gonna work that body. I'd be telling my professor. Other than calculus, which I think calculus still goes. Yeah, I think it's like 220 to 240. But I think it's the latest class. Well, I'm just saying in terms of afternoon, early afternoon. Because then
I guess like now you're ready for that 2 o'clock, so 4 o'clock.
well, that athletic reserve time. Oh, and there's also, I don't know what you're supposed to do. Like 4 o'clock. I think so. I thought about it now. It's like, okay, it sounds like I'm just doing
Luke, can you hear me all right?
Yes, I can.
How are you feeling?
I feel good. I feel really good today.
Good. Okay. Well, we look forward to seeing you back here next week then.
Yeah, this is not ideal.
Yeah. Just take the quiz from Mrs. Schaefer next week. Don't worry about the quiz.
Okay.
Oh, yeah. No, I got that. Oh, I know I did.
Like, okay, well.
No, like 36. Well.
No, and then I was like.
So that's where you're.
I wish I could.
I'm not an and just like reading those. And I just didn't really study them, so I was like. And it was like, yeah. And for real, like.
We're supposed to go to Clark Center's place tomorrow.
Now they're not.
Rain, snow, and ice. I can't see it. Remember Clark Center, we have all those off the road, those off the roads, then we have a cloud, or something.
Yeah.
Plus you got just 2 places in the code.
Oh, I told you, I was like, forget. Yeah.
So tired.
Okay, those of you in the second lab section, I'm just going to give these back.
I can read that.
Or is it right around the top of the locker?
I don't either, quite frankly. And I don't know, I mean, it just takes time, so like. To be very powerful.
Just pick up the quiz and just take it. I'm going to start, so. No, you didn't.
To be fair, we probably could have figured it out,
Yeah, but
but.
why guess? Okay, let's go ahead and get started. So we were talking last time about when the action potential gets to the axon terminus, the terminal bouton, as it's called, the terminal button, you activate a voltage-gated calcium channel. The voltage-gated calcium channel is going to cause exocytosis of those neurotransmitter-laden vesicles. So if it's a motor neuron, those neurotransmitters will be acetylcholine. If it's a neuron from the midbrain going up to the basal ganglia, the dopaminergic neuron, it'll be dopamine. If it is a learning and memory type neuron associated with the prefrontal cortex, it'll be acetylcholine as well. If it's mood-based, it'll be serotonin. If it's word-based, centers itself in the blah, 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 so on and so forth. So these will fuse with the terminal. There's a whole machinery that mediates that. That machinery are called snare proteins. Snares form complexes that mediate the fusion of the vesicles with the membrane and then the dumping of the neurotransmitters into this space between the two neurons. That space is called the synaptic cleft. Now, it's easy to think that the synaptic cleft is empty space. It is not. It's filled with a network of extracellular matrix proteins that are collectively known as the post-synaptic density. So the proteins in there are called PSD, and then usually it's according to their molecular weight. You see this dark stuff here? That's postsynaptic density. For years, it was thought that was just the fixation process caused the cells to get damaged, and that was just the cells kind of vomiting the results of the damage caused by the fixation. That turns out not to be the case. That's a real thing, and it turns out mutations in postsynaptic density can affect neurotransmission across neurons. So you have the space called the synaptic cleft. There's this stuff in there called the postsynaptic density that provides structure to that synapse. This shows that it's empty space, so that's not quite accurate. These neurotransmitters will bind ligand-gated channels in the membrane of the postsynaptic cell. So this is the presynaptic cell, this is the postsynaptic cell. Signaling goes this direction from the presynaptic cell to the postsynaptic cell. Some of those receptors that it will bind will be activated, and you'll get usually a potential, some sort of graded potential. Remember, graded potentials are localized, but they're additive. And the more of the neurotransmitter you get, the greater the graded potential that you get. Now, when after the neuron generates an action potential, it is in that state where it's hyperpolarized. So at that point, it can't start another action potential. And that period of time during which you cannot activate another action potential is called the refractory period. And refractory is a term that we use if someone is stubborn. You're so refractory to change and resistant to change. So the refractory period, the neuron stubbornly resists being activated again. So you have this so-called absolute refractory period, this time here, and that is about 1 millisecond. And then you have the so-called relative refractory period. It's called relative because this period of time can actually change. It can be shortened. And if you have neurons that need to fire repeatedly, this can be shortened, and you can actually fire again soon thereafter sometimes. So the absolute refractory period, though, is about 1 millisecond, after which you cannot mount another action potential because you're already in the midst of one, essentially. So you can't, the channels have, the sodium channels have their inactivation gates closed. You can't essentially open them anew. That wouldn't solve anything. Now, this is not the case for dendrites because the graded potentials are of a different quality than the all or nothing potentials that you get in the axons. So the bits of the nerve that receive the stimulus, the dendrite, the cell body, what have you, they get added up throughout the neuron. And because they get added up over the entire cell body of the neuron, you don't really have a threshold for graded potential, so there is no refractory period. And you can easily re-stimulate and re-stimulate and re-stimulate. However, for the all or nothing potential, you have a refractory period. That refractory period, as we showed you, is relative, 1 millisecond, and absolute, 1 millisecond, and relative, which here looks like it's about 2.7, 2 3/4 milliseconds. That can be shortened, however, to less if you need it to be shortened. So the usual speed of transmission is in the vicinity of 1 meter per second. That's going to vary according to the diameter of the axon. Larger axons will tend to transmit faster than smaller ones. So for example, there are aquatic invertebrates that have these feeding structures. And if they sense that danger is afoot, they'll retract those feeding structures really quickly. And so the faster they can retract those feeding structures, the more likely they are to survive. So we essentially have these axons in these particular animals that are really big. The typical run-of-the-mill nerve, whatever that means, and it's the kind of the average, given the average diameter of nerves in our body, 1 meter per second is a pretty good guess, pretty good average. Some nerves are coated with myelin. Myelin, because the transmission jumps from one node to another, that's going to speed things up greatly. When you coat an axon with myelin, you have a cell that undergoes a very specific attachment recognition between the axon and the cell. The cell then undergoes very profound cell shape changes. It flattens out from a from kind of a balloon to a piece of paper. This flattening is a really remarkable thing. In fact, you can actually take other cells and engineer them with some of the receptors and axons. And if they touch these precursor cells, they'll just flatten out like nobody's business. And then they start to make other attachment molecules that will cause the cell to attach to itself. And they just wrap, and they 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 form the so-called myelin sheath, which you can see is kind of an onion skin layer. And this forms a so-called sheath of Schwann. There are two different types of cells that do this. In the peripheral nervous system, this cell is called a Schwann cell. In the central nervous system, the cell is called an oligodendrocyte. Oligodendrocytes can wrap several axons. So one cell can do multiple axons. Schwann cells, just one cell wraps one axon. Schwann cells are kind of cool because you can, just with a little peripheral nerve biopsy, you can get these cells and you can culture them. They divide actually quite well in culture. And then you can actually use them to repair damaged myelinated nerves. They'll actually move in next to the nerve, and they'll recognize it, and they'll attach to it, and they'll wrap it. And that's a very exciting field of research that hopefully in your lifetimes, you'll see people getting treatments, off-the-shelf treatments to repair damaged nerves. So this occurs in the peripheral nervous system, And the Schwann cell wraps it and wraps it and wraps it. And here's a really beautiful picture again, shamelessly stolen from your book, that shows this onion skin type of wrapping, essentially, of the neuron. And here's a very, very high resolution, very high magnification picture. Boy, look at the regularity of that wrapping. Isn't that cool? It just looks like something done by a computer, doesn't it? And yet it's so regular. I find that really remarkable. So these express a lipoprotein called myelin, very, very high quantities. Myelin is what's responsible for the insulation of the nerve. Here's an oligodendrocyte. Notice that this oligodendrocyte takes on several axons at a time. That's a big difference between Schwann cells and oligodendrocytes. Oligodendrocytes act as precursors. They're called oligodendrocyte precursor cells. Those cells you can also grow in culture, and they can divide. You can inject them into the spinal cord, actually, for a spinal cord repair. And they will wrap several axons at a time. And the way they artist drew that, that looks like the eye of Sauron looking at you or something. They do have a big nucleus in the center. This is a type of glial cell, very important type of glial cell. Yes?
What role do oligodendrocytes and Schwann cells have when it comes to motor deficiencies?
Yeah, so multiple sclerosis is an autoimmune disease that attacks the myelin sheath in the central nervous system, and it damages the myelin sheath, and in some cases will kill off oligodendrocytes. And the treatments for multiple sclerosis are immune suppressive treatments to prevent the immune system from accessing the central nervous system and attacking the myelin sheath. And the way you diagnose it is with imaging scans of the brain that can detect demyelination that you can see with particular types of contrast and things like that. So the treatment that works the best is called Tyceba. Natalizumab is an antibody that binds to a integrin that lymphocytes need in order to get into the central nervous system. So that drug is by far the most effective has a big side effect called PML, peripheral myelin leukoencephalopathy, and it's due to a virus that everybody has called the John Campbell virus, JC virus. And if that virus is normally suppressed by our immune system, but if you're under these treatments, that virus can start to grow in your brain, and it can start to attack your brain cells and cause death. It can kill you, actually. So PML, you have to sample the cerebrospinal fluid for JC antigens. If you see it, you have to stop treatment right away and what have you. And it's, yeah. So that's how most of those treatments work. Damage to the spinal cord can actually kill them too. In fact, after a spinal cord injury, usually years after the injury, you're still killing off oligodendrocytes. That's why one of the proposed treatments is to take certain types of stem cells, differentiate them into OPCs, oligodendrocyte precursor cells, and inject them into the nerves and then have them myelinate the nerves and then try and see if you can get movement. Regenerating sensation seems to be easier than regenerating movement because the motor neurons, when they're damaged, they get severed, they retract, and then you make a scar, and that scar is full of proteoglycans, and the neuron can't go through it. So you have to try and find a way to get the motor neuron, extend its axon through that glial scar, which is full of proteoglycans and other things that cause it to retract, and then getting it to find the right place to the muscle, and then getting it to attach to the muscle, and getting the atrophied muscle back to where it's not a small thing. So that's hard to do. That's a field of science that is still in its infancy.
How do they check the status of like how in shape your myelin sheets are? Is there
Imaging. Yeah, imaging can tell if you have a lesion. The lesions have a different contrast in the background. I don't have time now, but I'll show you some. I've got some on some other slides that I show the nurses when we talk about. In fact, I just showed them Wednesday. You can see the lesions, they're pretty unmistakable. They just kind of jump out at you. So you use something called gadolinium, use a gadolinium contrast dye, and that they come up really well. All right, so the sheath, as you can see, is interrupted in places. Those are called nodes of Ranvier, and the action potential jumps from one node to another. So that gives you this really fast transmission through myelin sheaths. And that's necessary for certain types of nerves to be able to do what they do. And this is called saltatory transduction. And if your average rate of transmission is 1 meter per second, in the myelin sheath, it's 100 meters per second. So that shows you just how well myelination increases the rate of neurotransmission. So these are involved in quick stretch receptors, skin and skeletal muscles. Those of you who play certain sports where your limbs get stretched back, I'm thinking of bullies, where you might reach for a ball, and then all of a sudden your arm all of a sudden will just like a reflex. That's essentially your spindle tendon organs, essentially saying, up too far, you're going to damage yourself, and then you get a reflex that stretches forward, so you don't do that. In fact, sometimes a reflex can actually be quite painful, and that can actually hurt you. Baseball players, sometimes if you go too far, or if you catch a ball and it puts it way back, all of a sudden your arm will all of a sudden spring forward, or if you're throwing and you get it back too far, it'll actually cock up your throwing because you get this reflex and it's not controlled. It's just a reflex. And so the ball will just all of a sudden shoot out of your hand. You see that. I saw that once in a video I was watching. The guy, he caught the ball and then he put it back, and then the ball just literally fell out of his hand because it was a reflex thing. He got it back too far because the adrenaline was pumping, and he did that. And that part of your training is to get it back where you can get a good throw and not too far back. You get too far back, you're going to get a reflex. And those are myelinated axons. That's why you get that really fast sort of response. All right, so if we talk now about neurotransmitters, there are several different neurotransmitters, and some activate, some inhibit, some do both depending upon the receptor they bind. So that doesn't work. The best study of the neurotransmitters is acetylcholine. Acetylcholine is mostly an excitatory neurotransmitter. And we probably know it best from the fact that it binds to nicotinic receptors found in muscle, where it activates muscles. Nicotinic receptors are also found in the brain. And acetylcholine is an excitatory neurotransmitter in the brain as well. And acetylcholine plays a really important role in learning and memory as well. And in sleep and in lots of other things. Now, there are two different receptors for acetylcholine. There are nicotinic acetylcholine receptors. They're so called because they bind nicotine. And in fact, inhibiting these receptors is important in the control of smoking cessation. Most of the drugs that are used to control smoking are involved with acetylcholine reception. And then there are muscarinic acetylcholine receptors. These are very different receptors. Whereas this is a ligand-gated channel, these are so-called G-protein coupled receptors. Now they're called muscarinic receptors because they also bind these particular compounds that are produced by the mushroom Amanita muscaria. Amanita muscaria, it's a very hard mushroom to miss. The pileus, the cap, is red. I mean, here, see me, red. And yes, I found it out in the forest back here. When I was in high school, kids used to do shrooms. They'd take the cap and they would dry them. That concentrates the muscarinic acid, and then you just swallow them, and then all of a sudden everything becomes funny, and you have a nice little trip. And my brother, Pastor Tom, when he was in a rock band, he did shrooms several times, and he said it was very pleasant. And that's how they got their name. They bind these compounds. These compounds are hallucinogens because they essentially work through these particular receptors. So nicotinic receptors, they bind acetylcholine, they open, they let cations in. That depolarizes the cell with a gated potential. So these are ligand-gated channels. When you bind this channel, the effect is excitatory, almost always. Now, acetylcholine concentrations in the synaptic cleft is controlled by an enzyme that degrades it called acetylcholinesterase. This ensures that the activation of these receptors is not overdone. Now, think through this with me. Have any of you heard of a drug that's called Aricept? Anyone heard of Aricept? Any of you have elderly loved ones undergoing Alzheimer's? Anyone? No? Usually there's one in there. Well, okay, so the other one is Axel-1. These drugs are acetylcholinase inhibitors. So they increase the concentration of acetylcholine around neurons. That helps with short-term memory. And one of the first signs of Alzheimer's is a loss of short-term memory. And so these drugs are prescribed. This is donepezil, that's trade name of donepezil. This is the trade name of rivastigmine. And these are inhibitors of acetylcholinesterase. So you increase acetylcholine concentrations, increase learning and memory, essentially. Now they have side effects because cholinergics do lots of other things like give you diarrhea and can prevent, can decrease urinary incontinence and stuff like that. But for that, that's a small trade-off essentially for someone who's having trouble remembering things short term. So these are medications that you'll see given to people who show early signs of Alzheimer's. And this is how they work. They inhibit acetylcholinesterase. So you get more neurotransmission bang for your neurotransmitter buck, so to speak. So when a neurotransmitter binds to a receptor, and that manifests some sort of excitatory process in the neuron. That is called an excitatory postsynaptic potential. Excitatory postsynaptic potential. If acetylcholine binds to that receptor and excites it, creates an excitatory postsynaptic potential, because that's a bit of a mouthful, it's sometimes called an EPSP. The neurons that are excited by acetylcholine, the neurons that essentially get an EPSP from acetylcholine, they're called cholinergic. Acetylcholine activates them, so they're cholinergic. Neurons that are activated by dopamine are called dopaminergic. Neurons that are activated by serotonin are called serotonergic. Neurons that are activated by norepinephrine are called noradrenergic. So you see how this works. Now, your muscarinic receptors, if they're not ligand-gated channels, what are they? They're G protein coupled receptors. So these are proteins that are embedded in the membrane. They pass through the membrane 7 times. And then on the inside of the cell, there's a protein complex associated with them called the G protein. This G protein has 3 subunits. They're called alpha, beta, and gamma. Normally, they're associated with the receptor. But when the receptor binds its ligand, which in this case is acetylcholine, the G protein dissociates from the receptor. Now, there's another little trick with the G protein as well, and it's shown here. So these, you bind the ligand, these dissociate from the receptor because the receptor changes shape. These then go activate other targets. Now, the targets that these 2 subunits activate is this potassium channel. The potassium channel opens, potassium leaves the cell. Potassium leaves the cell. What does that do to the memory potential? Potassium leaves the cell. Positive charges are leaving the cell, so the memory potential becomes less positive. It becomes, it hyperpolarizes the cell. So this type of interaction is not excitatory, it's inhibitory. It's inhibitory. So if acetylcholine binds a nicotinic receptor, that's excitatory. You get an EPSP, excitatory postsynaptic potential. If it binds a muscarinic receptor like this, you hyperpolarize the cell. That's an inhibitory postsynaptic potential, or an IPSP. Same neurotransmitter, 2 completely different consequences. What's the difference? The receptor. Yeah.
So make it why is the excitatory and muscarinic still inhibitory?
Right, but there are some muscarinic receptors that are excitatory. Because there's several different types of muscarinic receptors, M1, M2, M3, M4, M5. And it depends. Several of the interactions are inhibitory. So for example, acetylcholine is responsible for slowing down the heart. That is an inhibitory reaction, albeit it's not with nerves, it's with muscles. Nerves that give cardiac muscle, acetylcholine. But some of these interactions with acetylcholine and muscarinic receptors are excitatory, some inhibitory. Depends on the receptor and it depends on the type of G protein that it's associated with. All right, so IPSPs, inhibitory postsynaptic potentials. These will hyperpolarize the cell. These will inhibit the cell. Excitatory postsynaptic potentials, these will activate the cell. They'll cause depolarizations of a gated potential type. Some other neurotransmitters are the monoamines. The monoamines include epinephrine, dopamine, and norepinephrine. That group is all part of the same biochemical pathway. You start with the amino acid tyrosine. The amino acid tyrosine is hydroxylated. using molecular oxygen. The enzyme is called tyrosine hydroxylase. It makes dopa, and then dopa decarboxylase converts it into norepinephrine, and then norepinephrine is methylated to make epinephrine. Because these essentially have as their parent compound catechol, they're called catecholamines. Catecholamines. Epinephrine, norepinephrine, dopamine, they're the catecholamines. They're synthesized from amino acid types. Serotonin is also a monoamine, but it is made from the amino acid tryptophan in a two-step biosynthetic process. No, sorry, four-step, my bad, four-step. These also bind to receptors, and the receptors they bind to are also G protein coupled receptors. Now, in this case, as you can see, the G protein is going to activate the downstream target, adenylate cyclase, which converts ATP into cyclic AMP. Cyclic AMP is a classic second messenger. Second messengers represent an amplification step. You activate one receptor, you make hundreds of these cyclic AMP molecules. These cyclic AMP molecules then activate thousands of these protein kinase molecules. Those thousands of protein kinase molecules will then affect tens of thousands, hundreds of thousands even, of these channels. So it's an amplification step. Cyclic AMP is a very commonly used second messenger. So by activating this G protein coupled receptor by norepinephrine, you make cyclic AMP that activates cell. This is an example of how you get an EPSP because you open that particular channel, and that turns out to be a cation channel that will let sodium ions into the cell. These neurotransmitters are also regulated in the synaptic cleft by an enzyme, two enzymes. One is called monoamine oxidase. It degrades monoamines. Then there's another enzyme called catechol methyltransferase, or COMT. Both of these regulate the concentration of these neurotransmitters within the synaptic cleft. And if you've ever heard of drugs called MAOIs, monoamine oxidase inhibitors, they are antidepressants. Not used that much anymore because of their side effect profiles, not terribly favorable, but they were very effective. They inhibit monoamine oxidase. They increase the concentration of monoamines. That increases your mood. COMT inhibitors. COMT inhibitors all end with the suffix capone. So think of Al Capone. So there's entacapone and faricapone. These are used for people with Parkinson's because COMT degrades dopamine, and by increasing dopamine levels, you decrease the symptoms of Parkinson's. And they're used in combination with other drugs because many times the drugs used for Parkinson's stop working. And so these are drugs essentially to augment add-ons for people with Parkinson's. Entacapone and entrecapone. And entrecapone. That's how you remember. That's my stupid mnemonic for the nursing students. I always have stupid mnemonics for them. And they giggle and they laugh and they say you're really dumb, but I think they appreciate them. So you see the action potential getting to the axon terminus. Calcium comes in via the voltage gated calcium channel. Calcium causes exocytosis. You fill the synaptic cleft with the monoamine neurotransmitter. You have a reuptake transporter that will get these back into the cell. You also have is inactivation by monoamine oxidase and also by the catechol methyltransferase. These will then bind to their receptors and usually elicit an excitatory postsynaptic potential. A cyclic AMP is one of them. Depends on the receptor. There's lots of different receptors for these. They whole classes of receptors. Serotonin. Serotonin also gets called 5-hydroxytryptamine. 5-hydroxytryptamine. So if you decarboxylate tryptophan, you make tryptamine. And then if you put a hydroxyl group on the indole ring of tryptophan, you make 5-hydroxytryptamine, otherwise known as serotonin. Serotonin is quite important for mood. And a very popular group of antidepressants are the serotonin selective reuptake inhibitors, or the SSRIs. So that includes such drugs as fluoxetine, trade name Prozac, paroxetine, trade name Paxil, citalopram, trade name Lexapro, escitalopram, trade name, I forgot, and other, do you remember escitalopram?
No, I just
Yeah, Lexapro. And then other drugs like that are used as antidepressants. They tend to increase your mood.
Hey, just a question, do we know the general name or do we have to know the scientific name for these?
Serotonin.
Okay.
You say serotonin, everyone knows.
I just
The
didn't know-
receptors for them are called 5-HT1A, 5-HT2A, 5-HT1D, 5-HT2B, 5-HT2C, 5-HT, blah, 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 you know, whatever. There's 14 different serotonin receptors, and they all do different things. So it's a bloody mess. So just remember serotonin has lots of different receptors. That's all you need to know. Yeah. The one they major on, they don't block the receptors. They block the reuptake of serotonin by the presynaptic cell. And that because the 1A receptor downregulates serotonin, they cause the neuron to downregulate its 1A receptors. So now the neuron inherently makes more serotonin. That's why it takes 2 to 5 weeks for those drugs to start working. The biggest problem with those drugs is that people stop taking them. Oh, it doesn't work. You have to give them time to work. And that's one of the caveats of those drugs.
So what's the difference between that and like an NDRI, which is like the
SNRI?
No, NDRI, which is like norepinephrine dopamine
Right.
reuptake. So like, what's the
Oh, NDRI, like bupropion.
yeah?
Yeah.
What's the difference? Like, how do they?
Those have dual action. Those have dual action.
What's that?
So SNRIs, for example, like venlafaxine,
Yeah.
at low dose, they prevent noradrenaline uptake. So you have more norepinephrine. Then at higher dose, they prevent serotonin uptake. So they can increase mood and treat depression, and depends. Venlafaxine gets used a lot, and venlafaxine.
Yeah.
That doesn't use a lot in veterans or PTSD.
Okay.
Bupropion, trade name Wellbutrin, or one that's used for smoking. Zyban.
I
Zyban.
thought bupropion was also used for smoking.
Zyban. Yeah, that's Zyban. That's a lower dose than Wellbutrin.
Okay.
Those essentially at one dose will prevent both dopamine and noradrenaline uptake. Yeah, that's a very clean drug. That's a good drug to add on to the drugs. Yeah.
So you say that it takes like 2 to 5 weeks to work. What happens if somebody takes it for a week and then stops taking it for a week?
It won't
And
work.
they just go, it just doesn't
It
work.
won't
It doesn't
work.
affect it in any
That's
other
why
way.
the psychiatrist has to tell you, don't stop taking this just because you think it's not working. It takes time to get it to work.
That doesn't have like any side effects by doing
If
it.
you, some fluoxetine has what's called a discontinuation effect, where you get really sleepy and then you might get hyperexcited. And yeah, that's an interesting side effect. That one is the worst because it has the longest half-life. Fluoxetine has a half-life of a week. That's why the drug is given once per week. Unless you have general anxiety disorder, in which case you take it every day. So, I'm really getting off track. This is not a farm class. I'm going to stop at this. All right, so serotonin is used a lot by what's called the WAIF nuclei. Do you have a question? No? Okay. I thought I saw your hand up out of the corner of my eye. Unless it was Elena just playing tricks on me. No, okay. Wasn't you, was it? No. Wasn't Kelsey playing tricks on me? No, okay. Sarah? No, not you. Oh, look at Sarah. Yes, Sarah, you're an angel. Yes. You never do anything wrong. Yes, really good kid. RAFE nuclei, yeah. RAFE nuclei play important roles in the regulation of mood and behavior and also alertness. And that's why SSRIs, one of the side effects is insomnia, because the RAFE nuclei need to be repressed in order for you to get to sleep. So here's essentially the serotonergic system. So here are the RAFE nuclei, but you see the RAFE nuclei extend their axons to lots of other places. So they extend their axons to basal ganglia, to the thalamus, and then throughout essentially the cerebral cortex. They're also involved to some extent with coordination as well, and what have you. They do a lot of different things. This set of neurons in here, this is called the reticular system. That's really important for alertness. And in fact, when you go to sleep, that's a system that has to be suppressed. And in fact, general anesthesia, that's how general anesthesia puts you to sleep. They just shut that whole thing down and you go to sleep, essentially. Dopamine is used by dopaminergic neurons. And there are 4 different main dopamine pathways in the brain. And the 2 main systems are the nigrostriatal dopamine system that starts in the midbrain, extends to the basal ganglia, the mesolimbic dopamine system, which starts right at the border between the forebrain and the midbrain at a place called the ventral tegmental area, the VTA. And then that extends, that's the system involved with rewards. So that's a system that is very active in people who are addicted to gambling or opiates or porn or whatever. So this is just taken from your book. There are 2 other dopamine systems too, not just these 2. So these are the main 2. So you can see the nigrostriatal system starts with a set of neurons in the midbrain called the substantia nigra. They then project, they're in the blue here, they project to the basal ganglia. Those are involved with voluntary movement. That's a system that goes south in Parkinson's disease. That's why Parkinson's patients will have the tremors and rest. That's why they have trouble initiating movement. That's why the movement will get the shuffle and the stooped sort of look like that. It can get worse as it progresses. The mesolimbic dopamine system will extend from the VTA essentially and then into the prefrontal cortex. And again, this is involved with motivation, reward, things like that. So Kelsey, just like that deep, deep pleasure you get from beating Iwu, essentially twice a day in a double header, that's the system. Okay. So addictive drugs essentially activate that dopaminergic pathway. And nicotine affects this pathway. They terminate in a portion of the brain called the nucleus accumbens. Nucleus accumbens is in the forebrain. That's the bit that this extends to. The nucleus accumbens is right here, essentially. And that lights up like a Christmas tree when you give an addict essentially what they want. Yeah. Norepinephrine is used, of course, by neurons of the sympathetic nervous system, so it gets used a lot in the peripheral nervous system. In the central nervous system, particularly in the forebrain, It's very important for mental arousal and focus. And this is a neurotransmitter that doesn't work, the transmission of it doesn't work well in people with ADHD. And most of the medications for ADHD increase norepinephrine neurotransmission in the forebrain. That's how they help you get focus and things like that. That's also why many people, adults with ADHD, will self-medicate with caffeine, because caffeine activates the same pathways that noradrenaline activate in the forebrain. And that's why the main treatments for ADHD are amphetamines, because amphetamines activate the noradrenergic neurons. They also cause increased heart rate, constriction of arteries, rise in blood pressure, and cocaine. Cocaine also activates this pathway, and that's why cocaine, you need more and more cocaine, but cocaine also activates the central nerve, the sympathetic nervous system. The blood pressure goes up, your heart rate goes up, and you have a heart attack with more and more cocaine. Two amino acids, glutamic acid, aspartic acid, these are excitatory neurotransmitters. In fact, glutamate is probably, it's fair to say, one of the main excitatory neurotransmitters in the brain. One particular glutaminergic receptor is called the NMDA, that's the N-methyl-D-alanine receptor. This particular receptor is extremely important in memory, and it's also very important in pain reception. That's why certain types of drugs that mitigate pain actually inhibit that receptor. So ketamine, for example, and also methadone, which not only inhibits mu receptors, but also inhibits NMDA receptors as well. Yeah.
So, like, at my job, we have like people who come and work with like to come off of like cocaine and stuff. How is that? How does that work?
Methadone is a very long half-life.
Okay.
So you get methadone, and the methadone will give you not quite the same high as the heroin does, but you get that quasi high, and it lasts for a long time. Now, methadone at those clinics, the only way you walk out with methadone is in your veins.
Right.
Yeah. So methadone comes in other formulations, but the only formulation that's administered is parenteral, is with a needle in your vein. So you walk away with the depot, and so the goal is to get you used to this kind of high as opposed to this kind of high. And then after the program, they start decreasing the amount of methadone so that you go from here to here, and then here, and then to here, and then to here. In England, the big problem they have is that people, because they're here, not here, they'll say, oh, I'll just top it off as a heroin. And they'll be. And then, of course, they blame the methadone. Yeah, of course, it's their fault. You sign all these papers when you do that sort of thing that says you can stay on this, you're committed to this. I think you have to be opiate free for 4 days before you can go on the program or something like that.
And there's not very many of them, right?
The efficacy of methadone has been debated.
Okay.
For some people, it definitely works. For others, it doesn't. And then, yeah, there've been some creative ways to people try to sell their methadone that they get. They actually will bleed themselves out, and they'll have people, wild stories. Glycine is an inhibitory neurotransmitter. Oh, yeah, Lena. The monosodium glutamate is for taste, of course, and glutamate is one of the things that binds a certain receptor in your taste buds called glutamate receptor, and that is a savory sort of thing. The thing with monosodium glutamate is that some people were having allergies to it, and that's why monosodium glutamate. Also, when you put monosodium glutamate in food, it checks up the sodium content, and so that was one of the issues. Lots of seasonings use it, but they'll use an alternative cation. And the executive seasoning, for example, that you can get for steak has monosodium glutamate, but it's monopotassium glutamate. which tastes just as good as one sort of make. And that was a big problem with it, is some people had allergies, either to that or to something else that was in that season. Oh, doesn't seem to affect neurotransmission. And that stuff has trouble penetrating the blood-brain barrier. Glutamate for neurotransmission is made inside the brain. Glycine is used in the peripheral nervous system. The peripheral nervous system. And it's used in the diaphragm to help relax. And strychnine actually blocks glycine receptors, antagonize glycine receptors, and you can't relax the diaphragm and you suffocate to death. And strychnine was used for a while as a rodent poison. I don't think it's used much anymore. In the central nervous system, the main inhibitory neurotransmitter is gamma-aminobutyric acid, or GABA. GABA will bind to the GABA receptor, and the GABA receptor is a ligand-gated chloride chain. So you get chloride essentially coming into the cell. That hyperpolarizes the cell, so it's inhibitory. And lots of neurons, a third of all neurons use GABA. Very, very widely used, and these are some of the neurons that die during Huntington's chorea, and that's what causes the uncontrolled movements associated with Huntington's chorea. There are also peptides that can serve as neurotransmitters. The ones that are the best worked on are the endogenous opioids. So these include endorphin, enkephalin, dynorphin, things like that. Endorphin is probably the one that's been studied the most. These are internal opiates that bind the opiate receptor, the mu, the kappa, the delta receptor. These are the receptors that also are antagonized by this drug, naloxone. This is the one that when given nasally, it's called Narcan. Narcan, nasal Narcan is carried by cops, firemen, EMTs. I think the trainers also have Narcan at the field house. You don't need a license to administer it. It's extremely easy to administer. You just spray it. Don't prime it. You prime it, you lace it. You just open it, put it in, it works, and it works fast. And then they don't wake up, you give the, because the packages come in tubes. You administer it, and then you put them in the recovery position. Recovery position. Yeah, that's recovery position. To keep them from swallowing spit and choking on it or something like that. Naloxone also comes in a tablet as well. So that blocks those receptors. Right. Now, Trexone is the long-lived one. That now naloxone doesn't have a long half-life. Now, Trexone is a long. That one's available usually just as a tablet. Gases can also act as neurotransmitters. Nitric oxide is used primarily in the peripheral nervous system. Carbon monoxide, very low levels of it, are used for odor adaptation in olfactory nerves. You must remember that neurons never have just one signal coming into them. They usually have several different signals coming into them, and so the neuron has to integrate inhibitory and excitatory signals and decide which one is the greatest. And if the excitatory signal gets the axon hillock above threshold, then the neuron will depolarize. So if you have enough of these excitatory postsynaptic potentials, you'll eventually get to threshold, and then you get the hillock rather to threshold, and then the axon will depolarize. Alternatively, that may not be the case. What if you have thrown in here an IPSP? Then maybe you'll go here, and you won't get up there. This phenomenon where you're integrating different signals coming in is called summation. And summation can be spatial. We have several presynaptic nerve fibers that converge on a single neuron, and lots of the membrane essentially has signals given to it so that you get the hillock above threshold. Spatial summation, or it could be temporal summation, where you have one maybe presynaptic neuron that just keeps firing. So over time, essentially your postsynaptic membrane gets above threshold, so the hillock then gets above threshold. There are two ways essentially to skin the threshold cat, if you will, spatially or temporally. So here essentially are two neurons, both of them give excitatory signals, and you get the hillock up to threshold. That'd be an example of spatial summation. But if either of those signals is inhibitory, then that might not be the case. So one might be inhibitory, the other might be excitatory. That's not going to do it. So the summation essentially in this case won't quite get you there. So here's the cat's wow of all this, is that the final effect that you get in that postsynaptic cell, it's more than the input of just one cell. When we draw pictures on the board, we have one neuron, one presynaptic neuron, one postsynaptic neuron, and that's almost not the case. Usually a postsynaptic neuron has several inputs coming in. So it's rather the integration of several inputs that will converge upon that one cell and are summed, either spatially or temporally or both. If you get an all or nothing action potential going down that axon, the depolarization has occurred, but the effect that that depolarization will have at the axon terminus, that can be dulled by something called presynaptic inhibition. You have inhibitory neurons that form connections with the axon. And those inhibitory connections can decrease the amplitude of the depolarization. Therefore, the amount of neurotransmitter the presynaptic neuron releases is not as much. So you can have axonic synapses where you get inhibitory neurons that will connect not to the cell body, not to the dendrite, but to the axon. This will not prevent an action potential from occurring in the axon. Rather, it dulls the effect here because you need to depolarize this here so that the calcium, both these calcium channels can open, right? They're not going to open nearly as much. The amount of neurotransmitter you release is not that impressive. And then you can also have postsynaptic inhibition as well, where you have these different connections as well. And this is a picture from your book showing you some of these. Please look at that. Okay, we finished that. Monday, we will start in on nervous system 2, and we'll look at the brain. Okay, Luke, get well. Have a good weekend. Don't go out driving this weekend. It looks like it's going to be icy. So,
Alrighty. See ya.
okay, take care, brother.